my name is Timothy Gallagher. I'm a postdoc in Caroline Stromberg's lab where I've been working on ways to automatically classify grass silica short cell phytoliths, or GSSCP. Phytoliths have great potential for paleobotanical research. They can be diagnostic of the taxonomic group to which they belong, and grasses in particular have very distinctive phytoliths. You can see a sample of GSSCP diversity here in situ on the left, and isolated from many of the same samples on the right. Subfamilies, tribes, genera, and individual species seem to have distinctive GSSCP characteristics, whether it be morphotype, size, or specific shape features. Fossil GSSCP have been described from various paleontological contexts back to the Albian. They have been collected and described now from every continent. If we can successfully classify fossil GSSCP and place them within the grass family, this will allow us to trace the biogeographical history of the family and infer changes in habitat and ecosystems over time. Most previous work on GSSCP classification has been done using a 2D approach, and many of these approaches have worked quite well. But the 2D methods are challenging to apply consistently across all morphotypes, and they have only been tested on a small subset of grass phytolith diversity. In order to capture the full 3D shape and size of GSSCP, we used fluorescent stains and confocal microscopy to image isolated phytoliths from leaf samples across the grass family and produce 3D surface models. We also observed in situ GSSCP for many of the same taxa to establish orientation. This allowed us to use a landmark approach to align the phytoliths to their homologous positions as they would each appear in the leaf. As of today, we have completed 4,074 3D phytoliths from all subfamilies and tribes and nearly every subtribe. Some of the amazing morphological diversity of GSSCP are shown here, located at the appropriate place on the grass phylogeny. So using ANOVA, we found significant shape differences between nearly all recognized GSSCP morphotypes. A linear discriminant function analysis also showed that morphotypes clustered together fairly well. On the right, you can see the results of an LDA for axial bilobate phytoliths from 6 different tribes. Even though they share the same morphotype, the GSSCP from each tribe can be distinguished from the others, although there was some overlap among the 3 bamboo subtribes. Overall accuracy rates were modest, ranging from 52% accuracy at the genus level to 79% accuracy at the subfamily level. Cohen's kappa statistic, which measures performance of the model, found the best models were actually at the tribal level. These first results are very promising and demonstrate that the method can be applied across a diverse set of morphotypes. We are confident that we can improve on these methods with better alignment strategies and increased sample sizes. Classification models were also applied to GSSCP from Eocene sediments from Nebraska in the USA and Anatolia, Turkey. Most of the Eocene GSSCP from both sites were classified as woody bamboos, with some distinctive Nebraska GSSCP classified as herbaceous bamboos. These results are preliminary since we have only 5 of the grass subfamilies represented in our data. We are very much looking forward to future analyses with the full comprehensive data set. If you have any questions or would like to collaborate to bring your phytolith work to the 3rd dimension, please reach out to either Caroline Stromberg or to myself, and thank you very much for listening to our talk.